All right, we'll carry on here. We're pretty much right on time here for our next presenter. The last topic here of the afternoon in the FCC Theater. Following up to weather and matching soil moisture condition and so on and fertility. This next topic is to micro or not to micro. Important topic. There's been more and more questions coming out about that in the last few years. Potentially less livestock, less manure applied. My yields are higher. Do I need to put more micros to the ground to supply my crops? So those are some of the questions we're likely going to have addressed here for you today. So for that, we have Dr. Jeff Schoenau, who is a professor with the Department of Soil Science at the University of Saskatchewan. Dr. Schoenau is a professor, professional agrologist who works with the Soil Science Department at the university. He holds the Saskatchewan Ministry of Agriculture Soil Nutrient Management Chair in the College of Agriculture and Bioresources, and is a fellow of the Agriculture Institute of Canada. He was born in Saskatchewan and completed his undergraduate and graduate degrees in the College of Agriculture at the University of Saskatchewan. His research, teaching, extension activities deal with soil fertility and fertilizer management, nutrient cycling, and practical approaches to soil conservation and improvement. Please welcome Dr. Jeff Schoenau.
Great. Thanks a lot, Regine. I'm really happy to be here in Manitoba this week to talk a little bit about, to answer the question, to micro or not micro. And indeed, that is the question, folks. It's not a real easy question to answer. And in fact, it's pretty complex, and I really hope I can shed some little bit of light on this mystery. But as always, in science, sometimes you end up with a lot more questions than you do answers. I'd really like to acknowledge the folks that have worked with me on micronutrients over the past years, some graduate students, research associates, who have worked along with me to address some of the questions about where might we expect to see a micronutrient deficiency, what kinds of things affect the responses that we're going to see, and kind of putting it all together. Certainly when we think about micronutrients, there's a reason why they're called micronutrients, folks. They're elements that are required in very small amounts. In fact, an example is copper, a micronutrient that's required in only a few grams per acre. But nonetheless, even though those elements we call micronutrients are required only in small amounts, they're just as important as the macronutrients we're most familiar with, like nitrogen, phosphorus, potassium, and sulfur. In Western Canada, the micronutrients that are of interest to us folks are elements copper, zinc, boron, manganese, iron, and chlorine. And those are the ones that I'm going to talk to you about this afternoon. There's a couple of others, nickel, molybdenum, but deficiencies of those micronutrients are essentially unheard of. And I like to think, micronutrients are a little bit mysterious in nature. I guess for lack of a better term, they're kind of ghostly. Ghostly because the deficiency symptoms sometimes appear and then sometimes unexpectedly disappear. And the other thing about them is they tend to be kind of ducking in and out of dark holes. The symptoms are often easily confused with other kinds of stresses out there, environmental stresses, and the symptoms they produce are often not that specific, depending on the nutrient element. And the other question that certainly comes up is, what kind of conditions am I likely to see these deficiencies show up? Oftentimes, folks, it takes a rather unique combination of soil, environmental, crop conditions in order for that micronutrient deficiency to show up. And finally, as I'll show you when I'm summarizing some of the research results, oftentimes it's kind of difficult to conclusively diagnose and predict a response. And that's because those responses are oftentimes small, fleeting, and variable. So it's kind of hard to get a handle on these micros from the outset, and that's what makes them challenging. A little bit of biochemistry, I think, is in order here to help understand availability of micronutrients. First thing is that micronutrient metals are typically taken up as cations. That's a positively charged ion. And folks, those cations tend to really strongly sorb onto soil particles. How the micronutrient metals actually get to the roots is through the formation of a chelate. The micronutrient metal binds with a ring-type structure, an organic molecule in solution, and then it can kind of roll along and move through the soil water to that root. So that chelation reaction is really important in helping those micronutrients stay in solution and allow them to be able to move to the root. In fact, some micronutrient fertilizer formulations are sold as chelates in order to help them become more available. In other words, help them move through the soil to that root. Boron and chlorine are a couple of other micronutrients. They're a little bit different. They tend to exist in the soil water, and they move along with that water wherever that water is moving. And as John mentioned, in terms of a movement process, we call that mass flow. And really it means that they're quite mobile in the soil. And a little bit of water in the soil can really influence the distribution of a mobile micronutrient like boron and chlorine. The uptake of these micronutrients, folks, occurs mainly through the roots, but we can also get uptake through the foliage, through the leaves. And that's why foliar application of micronutrient is another option in applying those micronutrient fertilizers. A little bit of physiology, I think, is also in order. The role of micronutrients in crop nutrition. The micronutrient elements, the metals, copper, zinc, manganese, and iron, I put those together, folks, because those are micronutrient elements that are oftentimes involved in, as well as photosynthesis, which is obviously very important to the plant, enzymatic reactions in the plant, activating enzymes, sometimes also involved in hormonal functions in the plant as well. And I think the interesting thing as well about some of these micronutrients is they can also have kind of indirect effects on plant growth through their role that they play in aspects of disease resistance. And a good example of that is copper. Copper deficiency may aggravate the incidence of ergot in cereals. The reason why is that the copper deficiency causes the floret of the cereal to remain open a little bit longer, a little bit wider, which increases the possibility of infection by the ergot organism. So some interesting interactions there between some of these micronutrients and diseases, folks. Boron. Boron is an element that's involved with cell division, a multiplication of cells, especially at the growing point of plants. And especially boron deficiency affects pollination. And that's because boron is a good example of a particular physiological role of boron is its role in the elongation of pollen tubes. And so without sufficient boron, you don't get this elongation of pollen tubes, you have problems with fertilization, and you end up with poor seed set and poor yield. Chloride acts differently again. Chloride as a micronutrient is very important in balancing the charge in the plant. Chlorine also seems to play a role in the plant in water relations and cell turgor. And chlorine is also an example of another micronutrient that interacts with incidence of disease. And chlorine deficiency has sometimes been linked to increased incidence of leaf spot and also root diseases in cereal crops. And I'll talk a little bit more about that later on. I could probably spend a couple hours showing you pictures of micronutrient deficiency symptoms. However, I don't think it'd probably really do a lot of good. And for a lot of the micronutrients, the deficiency symptoms are not that specific. But a couple of micronutrients that I do want to point out do produce rather specific symptoms. And one is copper deficiency in cereals. Copper deficiency in cereals produces a kind of a pinching off, a twisting, sometimes referred to as a pigtailing, a curling of the ends of the leaves, as shown here in wheat and also in canary seed. The other micronutrient deficiency I show you here is zinc deficient wheat. And I put this in here because you got a lot of green in there, but the one thing that you see is this kind of yellowish color in these wheat plants that are growing on a very zinc deficient soil. Zinc deficiency in a cereal will often show up as a yellowing, small leaves, sometimes malformed leaves, and sometimes if you get up close in a zinc deficiency, you will see the yellowing or the chlorosis appear in between the leaves. Now, here's some canola plants. They're obviously hurting units. They don't look too good. And actually, there's two different deficiencies at work here, despite the fact that they kind of look the same. Does anybody have any ideas of what kind of deficiency we might be looking at here in these two plants? Two different deficiencies. Well, that's sulfur deficiency in the canola, and we can see some kind of cupping and yellowing, especially on the young leaves. And we also see these small pods, poor seed set. What about this one here? Looks kind of like sulfur deficiency. Boron, that's right. Make it happen. Yeah, that's boron deficient canola. Kind of looks similar to sulfur deficient canola. Especially you've got the misshapen pods, poor seed set, a little bit of that yellowing and curling on the young parts of the plant. You can confuse things pretty easily sometimes in terms of different deficiencies because they sometimes have the same symptoms, or similar symptoms. And this is a good example. So folks, diagnosing micronutrient deficiencies on the basis of visual inspection, it may give you a good first clue depending on the micronutrient element, but it is risky and inconclusive. So what's your other options? Testing. Soil and tissue testing, folks, I think are useful tools. Although the tests for micronutrients certainly receive their fair share of debate and criticism. But I feel, having worked with both a lot of macronutrient and micronutrient testing, I think, as an attempt to measure what's biologically significant and important to the plant, I think those micronutrient tests probably aren't any better or worse than a lot of the macronutrient tests that are out there as well. And I guess one of the real challenging things with those micronutrients is the kind of variability that exists out there in the field that you're trying to account for. Folks, we need to recognize micronutrients can be very variable across farm fields, and that their deficiencies typically don't exist widespread across an entire field, but oftentimes are observed to occur in patches, localized areas, like eroded knolls within the field. And folks, in order to pick out whether you've got a limitation there, your sampling strategy has to be able to work to pick out those areas. Taking a bunch of cores across the entire field area and lumping them all together won't allow you to identify very well specific zones or patches in the field that indeed a micronutrient deficiency may be a limitation. So you really need to know where to look. So what kinds of soil conditions contribute to micronutrient deficiency? Well, one of the first ones to think about is the texture of the soil. And in particular, sandy soils are soils that are more prone to micronutrient deficiency because it's the clay-sized fraction that tends to contain a lot of those micronutrient elements like copper and zinc as a slow reserve, and also the clays can help hold on to those micronutrients in the soil. If the soil's real sandy, you don't have a lot of supply, a lot of release from weathering doesn't exist, and the sandy soil texture means that you don't have a great ability to hold on to them either and protect them from loss. Calcareous, high pH soils also tend to be candidates for increased incidence of micronutrient deficiency, especially micronutrients like zinc. In Manitoba here, as John said, soils here tend to be a fair bit more calcareous, more lime than what we would find in Saskatchewan or Alberta. And we know that that high lime content will fix some of those micronutrients, especially copper and zinc, into less soluble zinc carbonate and copper carbonate forms. And in fact, we've used the Canadian light source, the synchrotron in Saskatoon, to actually show that the fate of some fertilizer, copper and zinc, that's added to these calcareous soils is in fact a carbonate mineral that's formed. It's partially available to following crops, but the kinds of really high pH conditions tend to render it less soluble and less available. Very low or high organic matter content, also something to watch out for when looking for micronutrient deficiencies. Boron deficiency is most often associated with very low organic matter soils, oftentimes sandy as well. On the other hand, peaty soils, like what you might find, for example, in the Interlake region of Manitoba, some of those peaty soils can suffer from deficiencies of copper, zinc, manganese, because in those peats, there isn't a lot of minerals to release those copper, zinc, and manganese ions into the soil water. Similarly, even in some very heavily manured, this is heavily cattle manured soils, I know in Alberta, some of those soils have actually shown up to be copper deficient. But I would say, folks, in general, manure addition does tend to increase the content of available micronutrient in the soil. But some heavily cattle-manured soils, there have been instances where copper deficiency has shown up because the organic matter binds the copper. And this kind of gets into nutrient balances and interactions. Probably the best known interaction related to micronutrient availability is the phosphorus-zinc interaction. That is to say, folks, in soils that are marginal in zinc, for example, application of a high rate of phosphorus can induce a rather severe zinc deficiency because the phosphorus interferes with the copper and zinc uptake by the plant. Some of our recent research work has also indicated that the addition of copper and zinc to a phosphorus-deficient soil without any added phosphorus fertilizer can also have a negative interaction and reduce plant growth. So we need to be thinking about how the availability of one nutrient certainly can impact and alter the availability and uptake potential of other nutrients as well. I think as a kind of general statement, sandy, gray, peaty soils in general would be the soils that we would expect to have the greatest frequency of micronutrient deficiencies. Just to share with you a little bit of data showing you the effect of texture. This was on a group of soils from southern Saskatchewan looking at the relationship between sand content and available copper as measured through extraction and PRS probe, and showing very clearly here that as the sand content goes down, the available copper as measured by extraction or supply rate increases. So low sand content, in other words, higher clay content, better copper fertility. That being said, generalizations about where, a micronutrient deficiency might occur can be a little bit dangerous, folks. The soil and the tissue test, as I said, can add some resolution, but there continues to be debate about critical levels. And I guess that's not surprising. It's pretty hard to establish a cut line in the sand between a hard and defined line in the sand or boundary or demarcation line between deficiency and sufficiency when there's kind of all of these different variables coming into play. So rather than having a hard line between below this it's deficient, above that it's sufficient, there's a gray zone that we call marginal. And it just reflects the uncertainty in what that level will actually translate into in a response, given all the variables out there that are likely to influence the overall micronutrient availability in that soil. Folks, I think a combination of soil plus tissue testing and also running some test strips in that field is one of the most conclusive ways in order to determine if on that farm there's a pretty good likelihood of seeing a response to micronutrient fertilizer addition. I'd like to spend the rest of the time, folks, to talk a little bit about crop responses to micronutrient fertilization. I'm going to be leaning fairly heavily on work that's been done in Saskatchewan and some work that's been done most recently in my group, but I've tried to pick out of this soils that I think are most relevant to Manitoba, picking some soils out of the black and the gray soil climatic zones on the eastern side of the province, as well as some Manitoba soils as well. So to start this bit of a coverage, folks, I start out with copper. The reason I start out with copper because of all of the micronutrient elements out there, copper is probably the micronutrient that has shown up as most limiting in crop production, particularly for cereal crops, especially wheat. As you go further west into Alberta, there's even a greater incidence of copper deficiency. I would say as you would move towards Manitoba from Saskatchewan, maybe less incidence of copper deficiency, but again, still potential may exist for that. Kinds of symptoms, again, to watch for in your crop, these pinched ends of the leaves that are sometimes twisted or curled. A little bit later on in the life cycle of a cereal, you may see empty heads, a kind of melanosis or browning from a distance. Those are symptoms of copper deficiency in cereal. An example here of some response to copper fertilization that we saw at a site in east central Saskatchewan on a sandy gray luvasol. And folks, this is one of the few examples I ever saw of a field where copper deficiency seemed to exist pretty well throughout the entire field. That's rather unusual. Again, most of these microdeficiencies tend to show up in isolated areas or patches sometimes within a field, rather than being really widespread throughout the entire field. But this one was. And actually, we got a pretty good response to copper that was applied as a foliar application at the flag leaf. For whatever reason, the soil incorporation, this was work that was done by Dr. Molly at Agriculture Agri-Food Canada, the soil application really didn't produce much yield benefit. Kind of along those lines, Pat Flayton in Saskatchewan, following up, also did some work and found foliar application to be the most effective strategy for addressing copper deficiency in wheat in the year of application. Some of our work that follows up on this, I'll show you, actually showed that both foliar and copper, and soil-applied copper, I should say, foliar and soil-applied, both of those tended to work pretty well for alleviating copper deficiencies in wheat. So different conditions can affect the relative performance of a soil application versus a foliar application. Some work that we've been doing recently, and this is Dr. Ryan Hangs, who's been working with me, and we finished a project. We were interested not only in the response of crops grown in rotation to the added micronutrient that year, but we were also interested in how micronutrients might carry over in a rotation and affect the yield of subsequent crops that followed in rotation. So we did work in the glass house with copper, zinc, and boron in a wheat pea canola rotation. Now, we were thinking, do we do field trials and do maybe 4 or 5 field trials? Or would it be more efficient with the resources that we had to actually go out across Western Canada and get large amounts of soil from all 3 prairie provinces, 47 soils, and bring that soil back in and look at micronutrient response under controlled conditions? The challenge with doing field trial work with micronutrients is you need lots of replication, and because, again, of so many factors that can influence the response, you may do field trials at a site for 2 or 3 years, and because of the environmental conditions, not see a response at all. So this way we set it up. We went out, we got soil, large amounts out of the field, literally thousands of pounds. We brought them back in, mixed them up, and ultimately then put them in really big pots. And what did we find in this work? We had 12 mineral soils that, out of that group of 47 that were generally suspected as potentially being responsive to micronutrient fertilization based on soil test. So the first year we added copper to wheat. And of those 12 soils, we got some significant responses to both foliar applied copper as a chelator copper sulfate and also copper that was banded in the soil. Interestingly, in this study, the banded chelated form of copper actually gave us a little bit of a yield decrease. And the reason why is because in those pots, we couldn't get the copper far enough away from the roots, and we're actually seeing some toxicity from that form of copper. And that's the thing also to remember about with micronutrients, folks, is sometimes it's a pretty fine line, especially with copper and boron in particular, a fine line between sufficiency and toxicity. So you don't want to be putting this stuff on at really high rates year after year, especially close to the seed. And this, I think, is a good example. You want to be applying at recommended rates according to some type of guidance that comes from your diagnostic tools. Interestingly, the next year we grew peas on those soils, and we added some copper, or I should say we added some zinc to the peas. We didn't add any copper to the peas. But where we added the copper the year before to the wheat, we saw in the peas that were growing on that soil the following year a positive response of the peas to the copper that had been applied the year before to the wheat, which was really interesting. Don't really know the reason why. Maybe it was a nutritional thing. Maybe the copper, because it can act as a fungicide, was actually controlling some root rots in that soil. Anyway, a good example of carryover of a nutrient from one crop applied in one season to a different crop growing the next year that in this case produced kind of an unexpected, undocumented response. When it came to zinc, all of our different zinc treatments, as shown there, there was maybe a trend towards a yield increase, but overall, we didn't get a big response to the zinc that we applied to the peas, but we did from the copper applied the year before. Again, we don't know what the reason for this is. It's something I think we'd like to take a look at down the road. There was a trend to the pea yield responding positively to the zinc application, but it wasn't statistically significant. And folks, this is a story that you'll hear me sing over time with zinc, is that oftentimes in trials we see a trend towards a positive response, but it's really hard to show it as being statistically significant at the 5% level. And that's just because it seems like there's a lot of variability out there, a lot of different things affecting that response. Which moves me on to zinc. Going back a little bit into history, a number of trials conducted back in Saskatchewan in the 1980s with a number of small grain crops really didn't show any significant yield responses to zinc. We followed up a little bit on this, and we could show that some areas or patches within the field, eroded knolls, in fact, were responsive to zinc fertilization. We didn't do any work with corn, but important to recognize, folks, and I know a lot of corn's growing here in Manitoba, corn has a high zinc requirement, and response may be anticipated in low testing soils. And corn's also a high phosphorus user. And remember that high rates of phosphorus application to soils that are marginal or deficient in zinc can induce this zinc deficiency as a result of the phosphorus somehow reducing the uptake, the availability of the zinc. We did some work with lentils a few years back on some southern Saskatchewan soils. Again, what do we find? Variability in response to application of zinc fertilizer. We saw a few positive soil responses on soils from southwestern Saskatchewan, but as we moved to the north and east where the organic matter content was higher, the responses diminished. So finally, we didn't see any responses at all. And we did see, and this is an example here of a soil and Ardale association. You can see here the benefit from adding 2.5 kilograms of zinc sulfate per hectare, 5 kilograms of zinc sulfate per hectare on the height of that plant, the growth of that plant. But sometimes it does, sometimes it doesn't translate into a significant grain yield increase. We went out into the field and did some zinc fertilization responses with lentils. At one site, actually was a site on my own farm, very low testing in available zinc. We expected to see a yield response. Flat line. No response to zinc fertilization. It might have had something to do with how we applied the zinc. We applied it as a broadcast zinc sulfate. There's a lot of carbonates in that soil, and we know from our assessments of the zinc, the fate of the zinc in the soil, probably a lot of it did get fixed. Maybe we would have got response there if we had banded that zinc instead of broadcasting it on the surface. At the Saskatoon site, which had higher available zinc, we didn't expect to see a response, and we didn't see a response. So in that case, good agreement. We saw some trends for positive response of peas to zinc fertilization in the glass house, but it was only statistically significant, folks, for one of our organic soils. So this is the results of one of our glasshouse studies. This was a scepter soil, a soil of clay content. Pea response to zinc on this soil. The zinc level was fairly low, around that marginal level, 0.7 ppm. We expected to see a response. We did see a trend, but it wasn't statistically significant. It seemed like the soil application of zinc worked better. Interestingly, on this heavy clay soil, it tested quite high in available copper. So we didn't expect to see a response to copper fertilization, but actually we saw a significant yield response to the added copper. So again, your soil test, can give you an idea, but it's certainly not conclusive in terms of being able to predict a response alone. The one thing that I do want to point out in relation to zinc, in our studies, we not only looked at the yield response to added zinc of peas and lentils, but we also looked at how that zinc application affected the concentration of zinc in the grain. Why do we care about the concentration of zinc in the grain? Folks, the regions of the world that we sell our pulse crops, like the Middle East, Bangladesh, Pakistan, for example, a significant portion of their population suffers from zinc deficiency, human zinc deficiency. And so they like to buy pulse crops that are high in metals like zinc and iron that are important in human nutrition. So although growers don't necessarily get paid for that, it is an important consideration in the marketability of our Canadian pulse crops. And fertilization does increase that. I'll move on to boron. What crops do we tend to be most concerned about when it comes to boron deficiency? Canola, alfalfa are the crops that are most susceptible to boron deficiency. Reviewing the literature on this, folks, large yield responses to boron fertilization in Western Canada reported in the literature are rather rare. And even on soils, again, we come back to this, with low extractable available boron, where you might expect to see a response, some studies report no significant yield response of canola to added boron. In our work, kind of follows up along on this same finding. Only 2 of the 12 mineral soils that we've been working with responded positively to boron fertilization. So when I show this data here, response of canola to added boron on those 12 Saskatchewan mineral soils in our glasshouse study, compared to our unfertilized control, we're maybe seeing maybe a trend for some of those to be a little bit higher yield. We got boric acid, banded, foliar, and so on. But interestingly, this copper shows up again. In the third year after it was applied, this copper went on for the wheat. We grew peas, saw some positive benefit from the copper there. Now we're seeing a little bit of a positive benefit of that copper for the canola, the third year after application. Kind of interesting, something new, and something I think that's worthwhile following up. In terms of boron, 2 of the 12 mineral soils, canola did respond. Those soils were sandy, low organic matter soils, low in available boron, the kind of soil conditions where we would expect to see a response to boron, and we did. But interesting, that residual copper from 2015 producing that canola yield increase in 2017. One soil that did respond very positively in terms of canola yield to boron fertilization, soil-applied boron, was a White Fox Association Orthic dark gray Chernozem from the Nipawin area in the northeastern part of Saskatchewan, low testing in boron, and we did see a very significant yield response to soil-applied boron in that case. Manganese, cereals, especially oats growing on very peaty soils, could respond to manganese fertilization. But folks, I have to say, in terms of incidence of deficiency, manganese deficiency on mineral soils, very rare. But something to think a little bit about, manganese toxicity. Some work, an agronomist, Lyle Cowell with Nutrient in east central Saskatchewan, identifying some very sandy, acidic gray soils which were showing symptoms of manganese toxicity, probably due to very low pH and high soluble levels of manganese in the soil. So we took this soil, and we grew canola on this soil from east central Saskatchewan, very sandy, acidic, and we saw some symptoms that were rather characteristic of manganese toxicity. We see some yellowing around the edges of the leaves. The leaves of that canola had kind of a bubbly appearance. And we said, well, okay, if we increase the pH of that soil by liming, we should be able to reduce the soluble manganese in the soil and get rid of the toxicity and increase the yield. And yes, what we found was that when we limed that soil, the canola yield in those pots, the dry matter yield in the pots, doubled. And that was associated with a significant decrease in the tissue concentration of manganese below the toxic level. I don't know how widespread this is, folks. I think it's pretty isolated, but it's something to be aware of that could exist on very sandy, acidic soil. 2 in Manitoba. Okay, so something to watch out for. And speaking of Manitoba, I would be very remiss coming to Manitoba, where all the soybeans are growing, if I didn't talk a little bit about iron. And of course, iron deficiency produces rather marked symptoms. Iron deficiency chlorosis. IDC is shown here, that interveinal chlorosis that we see here, soybean plants at the tops of those plants. And there's a rather unique set of conditions that tends to really induce or bring on the onset of IDC. High pH, poor drainage, some heavy rains for example, high nitrates, high carbonates, a little bit of salinity aggravates iron deficiency. And in fact, soybeans, folks, are kind of inefficient users of iron to begin with. And of course, as you folks are aware, some varieties are more sensitive to iron deficiency chlorosis than others. And there are IDC ratings given for varieties. And I think they're really worth paying attention to in selecting a variety of soybeans if you're concerned about IDC on your own farm. We did a little study. One site, 2 years, so recognize its limitations. We did a field trial in southern Saskatchewan near Central Butte, 2015 and 2016, under soil conditions that were quite conducive to the development of IDC. High pH, slightly saline, high nitrate, lower slope. 2 soybean varieties in this trial. One was an IDC sensitive variety, Moosomin, and the other one was an IDC tolerant. What did we find? In 2015, that was a really dry year. And folks, we saw no response at all to any of our iron fertilization treatments. Soil applied, salt, chelate, foliar applied, nothing. But 2016 was a little different. That was a wet year. We got some rain in June and some flooding on that soil. And in that year, 2016, we saw that IDC-sensitive variety of soybean Moosomin respond significantly positively to a foliar application of iron. Whereas, not unexpectedly, the IDC-tolerant variety did not. It was unaffected by those conditions. And so this is our data here. That shows our control, our soil-applied iron, foliar-applied iron sulfate. There was maybe a trend there for significant yield increase for those two, but our best response came from our iron chelate that was foliar-applied. Again, one site, 2 years, so limited data set. Folks, I think genetics is your best defense when your iron deficiency chlorosis is a concern, but foliar iron might be a suitable rescue treatment. A lot of people talk about crops growing, or the soybeans growing out of the IDC. Indeed, that may well be the case. We did see a significant response at that site that year. Lastly, last one I want to talk a little bit about is chloride. Chloride, I think there is the potential for response of cereals in highly leached soils that have low extractable chloride levels. Particularly where chloride deficiency and response to chloride fertilization is showing up has been with cereals, especially winter cereals. A lot of work out of North Dakota, Pacific Northwest, indicating that some of that response as well may be associated with disease pressure. The ability of the chloride in some cases to reduce the infection of certain root rots in cereals. So again, some kind of complex factors coming into play related to the response. We did some work a number of years ago, landscape evaluation of response of wheat to application of 40 pounds per acre of potash. And we looked at responses in the depressions and on the tops of the hills. And interestingly in that work, 1 year, a year where there was a lot of snowmelt runoff and there was a lot of downward leaching, and remember chloride is mobile, it moves with water down, that leaching water removed the chloride from the soil profile. When we added some potash as a source of chloride, we saw a positive response. But just that 1 year. And folks, I think probably as more and more people are using potash, KCl in their blends, as John has talked about the importance of it, some responses to starter potash, I think that's probably likely reduced the incidence, the concern, the observance of chloride as a potential limitation in recent years. And I think the main reason why is in a plant, folks, most of the chloride stays behind in the straw rather than being removed in the grain. A lot of nutrients like nitrogen and phosphorus, 3/4 of the nitrogen and phosphorus in a mature plant is in the grain, which is removed. But in the case of chloride, almost all of it stays behind in the straw, which means it can keep recycling. Once you made that application of potash, you may be good to go for a number of years as far as not worrying about chloride deficiency showing up. Canary seed is a crop that has been shown, probably of all the crops, to be maybe most responsive to chloride addition. I had a fourth-year thesis student do a study in west central Saskatchewan. There was a small, but it was statistically significant response of canary seed to KCl. Bill May at Indian Head, Agriculture, Agri-Food Canada, showed seed yield increases of in his work, and I was involved with some of this, of upwards of 24%. From chloride added to canary seeds. So there is the potential there for some responses. Folks, as I wind to the end, some final thoughts. Micronutrients are something we can't forget about. Indeed, we need to be considering them as part of the overall balance. Balance is very important. Indeed, it's part of the whole 4R philosophy. The overall balance of nutrients required to optimize yield and economic return. And certainly we need to be thinking, I think, about micronutrients both in the short term, as in this season, but also over a number of years in the rotational cycle. And I think maybe thinking a little bit more, and maybe some of our recent research is pointing towards this, what are the potential benefits here to following crops in rotation? Not just that year, but the year beyond. Certainly micros do become a more important consideration when you're aiming for the top of the yield curve, when you're going for the maximum possible yield, trying to wring the maximum amount of yield you can get out of those genetics. The macros are all in order. That's maybe when you really start to think most intensely about those micros. They are patchy and they're variable, and folks, I think this really makes them an obvious candidate for a precision fertilization, identifying those micronutrient deficient areas within the field and addressing the deficiency in that area and not applying it to other areas of the field where it's not needed. I think the economics of that can look pretty good. Use a multiple evidence approach for identifying and verifying the deficiency. Because, as I stress again, there are so many complex factors that affect the availability and nutrition of those micronutrients that really makes that prediction of the likelihood of response challenging. And we've heard a lot about weather this afternoon and weather prediction. Good old crystal ball, right? Weather has a big impact. Environmental conditions are another set of complex factors that influence the availability of those micronutrients and likelihood of response. So I guess, you know, micronutrients are mysterious, as I say. They're kind of ghostly. Folks, I think we still have some more ghost busting to do in order to further improve those recommendations to you folks out there. And with that, I'll conclude, and thank you for the opportunity to come out of Saskatchewan to Manitoba. That's great, and I think we might have a few minutes for questions, Regine. Thank you.
Thank you very much, Dr. Schoenau, for a very enthusiastic and interesting presentation. Do we have questions from the audience? We have some time. Yes. Let me come up.
If a person's going to apply chelated zinc in wheat and boron in the canola, where would you get your best bang for your buck during herbicide application or fungicide application, or would it pay to apply both?
I think probably, at least from what we've seen, the application timing seems to be more important for boron in canola than it would be for addressing a zinc deficiency in wheat. You know, in our research work, and again, it's just our research work with a particular applicator system that we used. Interestingly, we had two different timings of application of foliar boron in our study versus a soil application. And unfortunately, in our study, neither of those foliar applications of boron worked very well. But I can think of other studies where foliar application has worked. But I think there's a more inconsistency and probably more effect of timing and how that relates to environmental conditions and movement of boron in the plant for boron compared to a zinc application. So probably your foliar zinc would be a little bit more consistent performer, is what I might predict.
At what time? Herbicide?
What time? And I have to admit, we did not look specifically at timings of application of zinc, but I would say probably and fungicide is going to come, your herbicide application is going to come before your fungicide application typically. I might lean a little bit towards the fungicide application in the boron, and maybe towards the fungicide if you're putting on like a flag leaf protectant for the zinc in the cereal. I don't think you want to get out there too early with those micronutrients as a foliar application, because their uptake actually tends to be more prolonged over the season, and sometimes some environmental conditions can actually interfere with the translocation, and if you've got it on too early, you might miss that. So yeah, I'm thinking, yeah, I would say, yeah, I would say probably, I would say probably closer to fungicide than herbicide.
Thank you. Other questions? Yes, just hang on.
What's your thoughts on Powerrich?
So Powerrich. Now, I'm not familiar with that particular product. I haven't worked with it. Does it have micros in it? Yeah. So again, I mean, thinking about fertilizer in general and inorganic fertilizers and macros and micros, again, you know, the balance is important. I guess the main thing, before you're paying for a micronutrient in a fertilizer, I think you want to make sure that, in fact, there is a likelihood of seeing a response to that micronutrient fertilizer on your own farm. So I would suggest even doing a little bit of testing before you kind of went with a multi-element product or a blend of a whole bunch of different products together to supply every nutrient element. You might want to do a little bit of investigative work beforehand to find out if some of those ones that you're putting on are, in fact, a limitation at all.
Thank you. Good question. Any other questions? Yes, one more.
I have heard of some folks putting on boron for heat stress in canola, even on crops that may not actually be experiencing any boron deficiency. Thoughts on that?
Yeah, and that's, I think, it's something with micros and even with some macros like calcium, we hear about, you know, applying those on, and really not from a nutritional standpoint, but as a way of alleviating some type of stress injury. Maybe it's heat, maybe it's hail. I've never done any work on that, so I can't really answer from that standpoint. I know what I've seen others report is some rather variable responses to that in terms of overcoming a stress. And it may be also related to the interaction of the micronutrient with hormones in the plant, but sort of related to a deficiency issue, not enough of it in the plant. I'm not sure whether there's a lot of evidence really in relation to some of the stresses. Not a big enough body of research work out there to probably provide a really strong answer to that question.
I think the batteries are out, and so okay. It's 4 o'clock. Last question. Going once, twice. Thank you for coming. Thank you to Dr. Schoenau. Let's give him a good hand of applause again.